Dear students, hope you all are fine. We were studying monosaccharides. We have already covered how pyranose and furanose ring, they are formed from open chain form of glucose. Now the ring form, that is pyranose and furanose, they are further of two types, alpha type and beta type. They differ in the orientation of hydrogen and hydroxyl group at carbon atom 1. So just see, this is carbon atom 1 of pyranose ring of glucose. If hydrogen is on the upper side, OH group is on the lower side, then this is the alpha form, this is alpha glucose. If at carbon atom 1, hydroxyl group is on the upper side, hydrogen is on the lower side, then this is the beta glucose. So alpha and beta forms which differ in the orientation of hydrogen and hydroxyl group at carbon atom 1, these are called anomers. Besides this, many sugars, they also rotate the plane of polarized light, either towards left or towards right. So if a sugar molecule rotates the plane of polarized light towards left, L stands for left, then it is known as levorotatory or L form of sugar. If it rotates the plane of polarized light towards right, then this is dextrorotatory or D form of the sugar. So D and L forms, they also differ in the orientation of hydrogen and hydroxyl group at particular carbon. For example, in case of glucose, D and L forms, they differ in the orientation of hydroxyl group and hydrogen group at carbon atom 5. If the glucose is towards the right side, then we say it is OH group is towards the right side, then this glucose is known as D glucose. If OH group is on the left side, the sugar is L type. So D and L form of glucose, they also differ in the orientation of hydroxyl group at carbon atom 5. Then DL mixture or racemic mixture is the mixture which contains equal proportion of D and L isomers. As D and L isomers, they are present in equal concentration, so this mixture will not show any optical activity. So it will be optically inactive, and the mixture is known as racemic mixture or DL mixture. Then there is one more term, epimers. When two monosaccharides, they differ in the orientation of hydrogen and hydroxyl group around a single carbon other than carbon atom 1. Carbon atom 1 is also known as anomeric carbon. Then these two sugars or two monosaccharides, they are known as the epimers. For example, glucose and galactose. This is the pyranose form of alpha glucose. This is the pyranose form of alpha galactose. Alpha because hydrogen is on the upper side, OH group is on the lower side. Glucose and galactose, they differ in orientation of hydrogen and hydroxyl group at carbon atom 4. So it means these are two monosaccharides which differ in the orientation of hydrogen and hydroxyl group at a carbon other than carbon atom 1. So glucose and galactose, they are known as the epimers. Now what are the chemical properties of monosaccharides? All the sugars which have free aldehyde or free ketone group, they can reduce. What is reduction? Reduction is the gain of electrons. So they can reduce cupric ions to cuprous ions, and hence they give reaction with Fehling solution and Benedict's reagent. These sugars which can reduce cupric ions to cuprous ions and give reaction with Fehling solution or Benedict's reagent, these are known as reducing sugars. So under monosaccharides, glucose, galactose, these are the reducing sugars. The aldehyde or ketone group of sugar, it can combine with OH group of other organic compound to form glycosidic bond. So bond formation will occur with the release of water molecule. So that's why this process is also known as condensation. So these are two chemical properties of monosaccharides. What are the functions of monosaccharides? Monosaccharides, as we have studied earlier also, they are produced as intermediates in photosynthesis. For example, a triose sugar glyceraldehyde, pentose sugar ribulose, then tetrose sugar erythrose, then haptose sugar pseudohaptulose, they are produced as intermediates in photosynthesis. Then glucose is the main source of fuel in the cells, we know. Then many monosaccharides, they are the component of other macromolecules. For example, ribose is a component of ribonucleic acid, RNA. adenosine triphosphate energy rich compound ATP, and coenzymes like NAD. NAD stands for nicotinamide adenine dinucleotide, and NADP nicotinamide adenine dinucleotide phosphate. We will study these coenzymes in another chapter. Deoxyribose is a component of DNA, that is deoxyribonucleic acid. Besides this, monosaccharides, they act as monomer unit for the building of oligosaccharides and polysaccharides. Besides this, glucose is also used in the formation of fats and amino acid. So these are functions of the monosaccharides. So under monosaccharides, we have studied 4 important terms. What are isomers? Isomers, these are the compounds having same molecular formula but different arrangement of atoms. So glucose, fructose and galactose, these are the isomers. Then what are epimers? Epimers, these are 2 monosaccharides which differ in orientation of hydrogen and hydroxyl group at a carbon other than carbon atom 1. So glucose and galactose, they are also known as epimers. Then sugar molecule, they can be, or glucose may be alpha form or beta form depending upon orientation of hydrogen and hydroxyl group at carbon atom 1, or they may be dextrorotatory or levorotatory depending upon the orientation of hydrogen and hydroxyl group at carbon atom 5. So this is all about monosaccharides. We will study in next video about oligosaccharides.